So today we are going to learn about the enzymes. So let's first define it because most of the time the question is about what are enzymes. It's a very direct question. How do we define it? Okay. Catalyst. You have missed one term. What is that term? Really? Oh, sorry. Let me 1 minute. Um, sure. It's a catalyst that speeds up the chemical reaction without being used up. There is one thing missing about like when we have to define the enzyme. What catalyst? What catalyst? What type of catalyst is this? Protein is made up of protein. So we call it a biocatalyst, or we can also say these are made up of protein. Okay, we can write here like this. Okay, what are proteins made up of? Amino acids. So there is a process in our body called as assimilation. What is this process? Good. The smaller molecules which are absorbed are then utilized by our body. Um, and their utility in the body. They combine up and make the bigger molecule. Okay. Next one is how do we name enzymes? Yes. What's their name end with? Sorry. How does their name end with? How does its name ends with A's? A's. Okay. Um, what? Why do we use? Okay, let's learn about some examples. These examples are important. Okay, with respect to all papers. What is the function of amylase? That is a function of saliva. Yes. It's break down starch into the smaller molecule, the maltose one. Okay. So starch doesn't directly break down into glucose. First of all, it's converted into maltose and then into glucose. Okay. What is the function of protease? It converts what? Proteins. It would work on protein and convert it into amino acids. What is the function of lipase? Fats into or lipids into glycerol and fatty acid. Okay. Then the next one is let's talk about the other one. There is one enzyme known as dehydrogenase. What does it do? Dehydrogenase. Okay. It would remove the hydrogen part. Okay. The name is quite simple because there are many molecules in our body that contains the hydrogen part. It removes that. Okay. Then there is another enzyme, catalase. This is another important one. So it's yes. So the function of catalase is to break down hydrogen peroxide into water and oxygen. Okay. The formula for hydrogen peroxide is H2O2. Okay. Okay. Next one is lactase that you're going to learn in the last chapter, the biotechnology one. What is the function of lactase? There are some people who cannot digest lactose sugar that is present in many lactose into glucose and galactose. Okay, that's why the thing that are usually made for the lactose intolerant people are sweet in taste because they would have simple sugars. Okay, lactose is a let's say a disaccharide, so it decompose into a simple sugar, a monosaccharide. catalase is an enzyme and it acts on hydrogen peroxide. These are the name of enzymes. So later we are going to discuss. Wait. Then there is one more which is important with respect to your exam. Pactinase. It would act on exactly fruit peel, and it convert it into a simpler unit, and it is used in the production of juices. Okay, now I have a question for you, or we can discuss this later in juices. We will discuss it later. Okay, there are some terms which are very important when we discuss enzymes. Let's talk about the important terms. One is substrate. What is substrate? Okay, on which the enzymes act is called as substrate. Then it further bind to enzyme and form a substrate enzyme complex. Okay. Okay, what is an active site? A place where substrate bind. Good. What is product? Chemical reaction. Good. Okay. So we say that these one, the first one that we have written, these all are enzymes. Okay. These ones are the substrates. Substrate, sorry, I missed one is. And these ones are products. So you have to learn this table. It might come in your MCQs part and the other questions part, that where you have to identify what is the product, what is this, or what is the correct product, or what is the correct substrate, or a correct enzyme. Okay, so we should know these names. Okay, then there is one lock and key model that defines, oh sorry, lock and key model that defines how enzymes work. So there are again 5 to 6 steps that you need to learn. आपको मैंने हमेशा बताया है कि whenever you are going to learn about something, you need to like make points of it. That I have to write 5 points for this one. Let's say we are going to write 5 points for this one. Let's write those 5 points. Number one, the number one point should be about the substrate and active site of an enzyme. Okay, so we say that the substrate arrange itself closer to the active site. Okay, then we also have to mention that substrate has a complementary shape to active site. Third point should be about binding. The substrate and enzyme bind and they form substrate enzyme complex. Okay, because I have to rephrase this one, so you need to have really have a good understanding of this one. Okay, number four. After binding, a chemical reaction take place. Okay, the products are formed. The products get detached from the active site because they are different. Now they are not complementary to the active site. They are the different compounds. Get detached, or what I have said, as they are not complementary to active site. What is the meaning of complementary? Exactly. That they can easily make a bond or they easily fit into each other. Okay. So these are the 5 points that you need to learn. Okay. Now the question won't be like this that you have to explain the enzymes work. The question would be like this. If explain the function of amylase according to lock and key model. How are you going to explain this? How? Then we won't say substrate. No, we won't say substrate. We have to name the substrate. What is a substrate? No, amylase is the enzyme. What is a substrate for amylase? Here, starch. Okay. So we say Protein. Yes. So we would write here substrate, right? The amylase, I guess color has changed. Yes, we have to name whatever substrate is here. We will write here amylase. Amylase would come near to the active site of, oh, sorry, I have to write starch. Starch would be near the active site of amylase. Okay. Then amylase has a complementary shape to the active site of amylase. Okay. It will form what enzyme substrate complex. There is no need to change it. A chemical reaction takes place, and what products are produced? Maltose or glucose? Glucose. Glucose is the final product. Maltose is the middle product. Hmm. No, no, we did here. Starch is the substrate. Okay. So amylase and starch, they have complementary shape. Okay. Starch. Yes. Wait. That is for the complex one. Let me rewrite it for you. Oh, sorry. Okay. So substrate is basically starch, right? And enzyme is amylase. Now, when I have written substrate has complementary shape to active site, then I have to tell the starch and amylase would have, or the active site of amylase would have complementary shape. So they get fixed into each other. Okay. So binding would take place and substrate enzyme complexes formed. Chemical reaction take place. As a result, glucose is produced, which get detached as they're not complementary to active site. Okay. This is how you explain the answer. So if the question is about protease, how you are going to tell this? Hmm. Protein. Exactly. Hmm. What substrate? What substrate? Protein has a complementary shape to the active site of protease. Okay. And there will be formation of substrate enzyme complex. Amino acids. Good. 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 Okay, so keep in mind whenever there is a question, okay, you would have a question of like 6 mark or 8 mark or 4 mark, then you have to arrange your answer. Okay, don't jump to the answer. Just write, first of all, write all the points that you need to mention in the answer. Okay, this is point number one, this is point number two, this is point number three, this is point number four. Only then you'll be able to give proper answer. It won't be off. It won't be off. So, exactly. And then you can also tell this at the end. Now the active site is available for further reaction. Okay, act because it gets detached, so active site is available for further reaction. Okay, till here. Now there are two important factors which are very important, which affects the enzyme activity. First factor is pH, and the second factor is temperature. So, okay, there are two questions related to it. Now you have to. I'm going to give answer for both. Let me use another one. Okay. First part is what happened to an enzyme when the temperature increases from 25 degrees Celsius to 35 degrees Celsius. Okay, this is your first part. Now, your second part would be what happened. No. What happens to the enzyme activity? Let's write activity so that it would be more clear. When the temperature increases from 35 degrees Celsius to 55 degrees Celsius. This was the exact question that was in the past paper. Activity, like consider it like for 6 mark. Okay, and how are we going to answer for first one?